From brain recovery and sleep research to epilepsy in tamariki Māori, nearly 100 neurological research projects have been disrupted due to COVID. 1 in 5 New Zealanders are affected by conditions like dementia, stroke, epilepsy, Parkinson's, brain injury, and over 700 more such conditions. And so far the pandemic has disrupted researchers working on preventions, treatments and cures in 92 different projects, including brain cancers, epilepsy and traumatic brain injuries. Some researchers have lost a year's worth of lab work. The Neurological Foundation has come up with over $1.2 million to support researchers during the lockdowns and to provide funds to get those projects back up and running. With us are 2 researchers who know this pain well. Dr Thomas Park is researching glioblastoma, an aggressive cancer that occurs in the brain or spinal cord, and Dr Deidre Jansson is researching the impact of poor sleep on brain repair and recovery. Kia ora korua, welcome both to 9 to Noon.
Good morning.
Good morning, kia ora.
Thanks very much. Thomas, could I begin with you explaining your research in the first instance, please, into glioblastoma? This is an aggressive cancer of the brain or spinal cord. Could you tell me a little bit about what you're working on?
Yes, indeed. First of all, thank you for having me on your program. And basically, as you mentioned, glioblastoma is the most highest grade brain tumor in adult humans. And it's the most lethal as well. And unfortunately, there's no cures for these tumors. And there's really a global push, which we are a part of, to find new therapies for this disease. And that's sort of what entails the research. And we really conduct this at the Centre for Brain Research at the University of Auckland, where we're really lucky to collaborate with neurosurgeons from Auckland Hospital that operate on these patients. And with their kind consent, we do research on the actual tumor specimens that are excised from the patients to come up with new therapies and to find out why these diseases occur in the first place.
What can you learn from that tissue? What theoretical treatments are you envisaging?
Right. Well, a lot of the research until more recently have been conducted largely on animals. And although they provide great insight into how the body mechanism and the tumors might work, because we're working on actual patient specimens, and humans are obviously different from other mammals. And these tumors behave differently to what we have seen also in animals. So we have this unique advantage where we can actually look at novel drugs, and we work with clinicians and chemists that can come up with these novel therapies, and also bioengineers to really tackle the intricacies and the information that's behind deciphering these tumors from actual patients. And because every patient is different, we're also able to see how different patients might respond to the same therapy. So that's sort of the research that we are undertaking at the moment.
And what was the impact of COVID?
Well, like with many industries, as you mentioned, it was a massive impact. One, because as I just mentioned, we need to keep the patient specimens that we've collected alive to do our experiments. And obviously during lockdown, where we could not access the lab at all, we had to either freeze or stop a lot of these cultures from going. And also due to the uncertain nature of the disease, we didn't quite know how COVID spread. And also we didn't want to burden the medical system, our medical colleagues. So we actually halted the collection of these specimens for over 2 years. It's now just about to start again, but this really did hamper a lot of the progress using these specimens. So we had to pivot a lot of our projects into more computational and database studies during the COVID times.
So did this mean that you actually effectively lost that 12 months of lab work?
Oh, yes. We probably will say 12 months or more of lab work, because it's not just about the time lost, but it's the stopping and restarting of these experiments. So yes, I would say at least 12 months.
So can you stop them and restart them, or have you lost some of what went before?
Oh, we'll lose some that went before. So there are parts of experiments where we can stop and start, but we're never sure if we stop it and freeze it and then bring it back out, whether they would still be the same as the original patient specimens. So we'll never know that. But that's what we have done with some of the experiments. But especially not just the actual research, but I feel really bad for the graduate students that were on these projects, normally graduate projects are about a year or 2 or 3 years. So to lose a whole year or more has been really devastating for them. But so I really feel bad for them, but they've done really well with the pivoted projects. But that's also been a intangible sort of impact on the progress of our work.
So you pivoted and did the more computational side of things. Was this work that would have happened eventually? Has it perhaps been brought forward suboptimally? What's the relationship between the lab work and the computations? Which comes first?
Yeah. No, actually, that's a bit of a silver lining, really. You try to take what you can do and look at the positive sides of things. And it was a project in planning, and as you know, with the transformation of the information era and information technology, it's the computational side's really really rising to the occasion. And we've done similar things where we really ramped the collaborations with the chemists and also the bioengineers to really look at the computational side of the project. And it's actually given us unique insights into the disease, which we're really excited to now pursue further once the specimens come back in, which will happen really soon. So it's one side of COVID that actually gave us new light, shall we say.
The thing is, with research, there's always funding, and the funding system in New Zealand is challenging. And so how was your research, and how is your research currently being funded?
Right. So as you are aware, and that's why me and Deidre are here today, is Neurological has funded a lot of our research. So as a sort of early career researcher, you start off with smaller grants and you start building up to larger ones based on your previous findings. And Neurological has really supported all that throughout my last 5 to 6 years of my research. So that's been really well funded. But because of this halting, the next funding round, you need to have results from your previous projects, which was delayed for about a year or two. So this sort of COVID relief fund, shall we say, from the Neurological has really helped continue our projects that had been delayed, and allows career researchers like me to really continue the work and be employed in this research as a neuroscientist.
However, can you get extensions to your grants? In other words, can you recover your funding track just as you try and get the research back on track?
Right. Yes. I mean, that's what the Neurological Foundation has provided. They've provided a whole year extension of funding, which is not a small amount because you have to include salary as well as the research costs. And they've also extended all my PhD students' scholarships as well for that amount of time, which has been fantastic. And I don't know how we would continue this research without it. So that's really been provided by the Neurological Foundation, and also the university has been really supportive, and they've allowed for funds to really bridge that gap. So that's what we are on right now until the next funding round.
I hear the coping mechanisms, and they're always needed, but how frustrating has this been?
I mean, it's frustrating, but you have to take it into context with the wider context. I mean, the whole world is going through this pandemic. And I mean, it's been frustrating, but I feel fortunate that I can still continue this research. And there are people that are suffering much more than we are. So we have to take it into the whole perspective. Yes, it has delayed our research, and we would love to find more, do research to find new cures or avenues to help the patients that are suffering from these diseases. But at the same time, we're really grateful and fortunate to be able to continue the research in the first place.
Ngā mihi, Thomas. Let's bring in Deidre Jansson. Welcome, Deidre. And can you tell me a bit about your situation? I think you were on a Wrightson Fellowship overseas when COVID hit.
Yeah, Kathryn, I was. So thanks for having me on. But so I traveled to Seattle with the Neurological Foundation Wrightson Fellowship, which was an amazing experience and opportunity. I went there to study in a world-class institute with internationally recognized clinicians and researchers. And then COVID hit, so it was quite hard because yeah, a lot of the stuff that I went to do there was to pursue mostly translational research. So to be able to do things and partake in studies that were directly related to patients and the disease and the things that they're going through. But because of COVID, essentially all the human studies in the institute where I was were put on hold and are only just starting to get up and running now, actually.
So what did that mean for you? Did you stay? Did you come home? What happened?
Yeah, so it was really hard. I had a unique experience, I think, because I went there while my family, so partner and children, stayed here so that I could go and get this amazing training and then return to New Zealand with all the experience that I'd gained, with the idea that I would travel back and forth, every couple of months. But because of COVID and restrictions, that ended up being only once a year, really. And then upon return, it kind of got really messy. Luckily, the Neurological Foundation extended my fellowship for 3 months. So I was able to get a little bit more done in the lab before I left. And then I was awarded a Neurological Foundation fellowship to return and continue my research here. But just the trying to get home with the MIQ and the possibility of my visa expiring and me being stuck in the United States without a visa was quite stressful. I was very lucky to actually get an MIQ spot and was able to return in time, but it was quite a stressful time, actually.
Specifically, what is your research about? This is the importance of sleep, but on brain repair, yeah?
Yeah, so my research is focused particularly on the regulation of cerebral spinal fluid. So we usually know cerebral spinal fluid, or CSF, to be something that surrounds the brain and acts as a cushion. But we're learning a lot more about what it does, and that it's actually important for sort of movement and type of cleansing of the brain. And in particular, this cleansing process that happens predominantly during sleep. So when we sleep, the brain essentially cleans itself. And in sleep disorders or instances like sleep apnea or sleep disruption, chronic sleep disruption, that process is impaired. So you sort of get a buildup of things in the brain, potentially toxic proteins that shouldn't be there, which could contribute to long-term disease, such as dementia and Alzheimer's related diseases.
It's incredible, isn't it, what we're learning, that this fluid, it's actually almost like a bath. I mean, it is literally cleansing away waste that is unhelpful.
Yeah, and it's amazing how much, we didn't know about it. Like this process was only recently discovered in the last 10 or 11 years. But just learning more and more about how it happens and how it's regulated. So it's influenced by things that would affect your blood vessels. So vascular disease, things like hypertension and stroke can also impact this process, as well as just overall like metabolism and exercise, as well as sleep. Like sleep is a big factor and something that I think is very underappreciated. We always say, oh yeah, we'll sleep later. But actually getting enough sleep is really critical in your brain's ability to function properly.
Do we know at what phase of sleep or in what phases of sleep this cleansing process happens?
Yeah, so it's predominantly happening during slow wave sleep. So in the course of an evening, you go through several sleep cycles that are composed of kind of REM or rapid eye movement sleep. And then there's a few different phases of non-REM, so non-rapid eye movement sleep. And this is where you get slow wave sleep, where you almost get like a synchronous activity throughout the brain. And this is usually where the cleansing happens. So researchers are trying to figure out particularly why this happens during slow wave sleep, and if there's other things that we can do so people that for whatever reason don't get enough slow wave sleep or enough sleep at all, if there's a way that we can sort of mimic what's happening in the brain during slow wave sleep to be able to promote this cleansing process.
What is being cleaned away? You mentioned proteins. What are the other waste products that are likely being cleaned away?
Yeah, so there's a lot of things. I don't think we know everything, but so essentially, metabolic processes or metabolic proteins and waste products. So when your neurons are active, they make waste just like almost everything else, and that's cleared. But the one thing that's particularly important is a protein called amyloid beta or beta amyloid, and this is something that we know to accumulate in the brains of people with Alzheimer's disease. And this is one such protein that is actually cleared by this process during sleep. And there's actually a study that was done that shows that just in humans, a single night of sleep deprivation causes an impairment of this clearance process, and like more amyloid to actually be found within the brain and within the cerebral spinal fluid just from a single night.
There's an irony to this, isn't it, in that the neurological effects of COVID and long COVID are going to be one of the many, many factors being studied going into the future. And here you are, each of you, with quite specific work, of course, on existing issues that has been impaired, and the Neurological Foundation helping out to keep you going. But it's no doubt it's an area, along with others, heart disease and others, that we are going to be researching during and post this pandemic. Tell me, Deidre, what's happened since you've come home then? How have you recalibrated what your research track was?
Yeah, so I actually, so I've been back in New Zealand since just before Christmas. And am now, I'm taking what I learned in the States about cerebral spinal fluid and sleep and how it affects CSF movement. And I'm applying that in the field of critical illness. So a lot of people that suffer critical illness actually also suffer sleep disruption and sleep fragmentation while they're in the ICU environment. And then between 50 to 80% of those people, even after they're released from hospital, will go on to develop some sort of cognitive impairment. So right now I'm focusing on trying to understand the role of sleep disruption in that development of cognitive impairment. And the reason that I've been able to do that is because I've been awarded a neurological fellowship to pursue this research. So they were just amazing and helping me to get back to New Zealand and continue my research, but also supporting just my travel to get back to New Zealand and the MIQ stuff. So that's what I'm able to do this simply because I've been supported by the Neurological Foundation. So
the funding's been extended, as it was with Thomas. It's such an important area, and just listening to you, at the time you most need to repair, right? You've been in a critical situation if you're in ICU. The time you most need to repair, your body's had a major insult or a major trauma. The sleep interruption as part of that and part of the treatment is hindering that repair. It's like a vicious circle, isn't it?
Yeah. It's really terrible. And I think researchers are starting to understand a little bit more about the importance of sleep. So there are a few research groups across the world. There's a few in the States that are particularly looking at this and the role of sleep disruption. But like I said before, because in the ICU environment is quite hard. You need to check on patients to make sure, they're doing well and surviving actually. So sort of getting around disrupting their sleep is quite difficult. So trying to find ways that we can mimic what sleep is doing so that we can hopefully promote the repair process is really a focus of a lot of research groups right now.
Apart from the interruptions, the sheer shock to the body and the brain of a major trauma, however it's happened, one suspects might interfere with so-called normal sleep anyway. I'm really interested in what those sleep mimicking processes might be when you hypothesize, when you look out and say, what is it we might be able to do that would almost trick the brain into thinking it was in a sleep state? What sorts of ideas do you have already?
So there's a few things. So one of the things that we know is that this clearance process that happens during sleep is partially regulated by like the movement of blood vessels. So like the dilation and contraction of blood vessels. So there are some drugs actually that can somewhat mimic this process. So some anesthesias in particular can mimic this process and potentially promote the cleansing. There's also some groups that are using a form of transcranial magnetic or transcranial electric stimulation to also be able to mimic the synchronous neuronal activity. So the way that the brain cells kind of act together during slow wave sleep, to be able to, again, mimic the process that happens during sleep so that you can promote the cleansing of the cerebrospinal fluid.
It would be massive. All power to you, both of you. Keep up your good work. I'm glad the Lifeline came. I know how stressful it is always trying to manage funding and manage research projects. Thank you both. Deidre Jansson speaking there, and Thomas Park.